So this slide should have been with the previous video, but it got lost somewhere. So anyway, there are several checkpoints in the process of, or in the life of a cell that directly affect mitosis. So all of these transitions between the different phases within interphase and then leading up to mitosis are regulated by special processes that evaluate whether or not the cell is ready to move on. So these checkpoints are really important in making sure that the cell is ready to move on to the next phase and then eventually into mitosis. So even though they don't happen in mitosis themselves, there are two checkpoints that happen within the interphase part of cell division. I'm sorry, of the cell life cycle. The first important one is called the start checkpoint. Now, the start checkpoint is the checkpoint that's going to say, okay, the cell is healthy and ready to start synthesis of DNA in preparation for mitosis. So this checkpoint after G1, between G1 and S phase, is going to evaluate whether or not the growth process of the cell has been completed, and whether or not the cell is ready to begin the process of preparing for cell division. If the cell doesn't pass this checkpoint, it's going to go into G0, or it's going to remain in G1 until everything is finished up and ready to go, and the cell reevaluates whether or not it's ready to move on to S phase. So then in S phase, DNA replication occurs, and then we have a second checkpoint in preparation for mitosis. And that checkpoint evaluates if DNA replication has finished, if there's any damage in the DNA molecules in the chromosomes that need to be repaired. So this is when your DNA proofreading enzymes are going to be working, if you have any double strand breaks, if you've broken any pieces of chromosome, those are going to be repaired. They're going to be reattached where they need to go. So then if your DNA is all ready to go, replication and repair has been completed, we are going to move into G2 and begin the process of mitosis. Once inside of mitosis, we have another checkpoint that's going to check whether or not those spindle fibers have been produced, that your centrioles and centrosomes here your asters have been produced, your microtubules have grown, and then that checkpoint is going to make sure that all of your chromosomes are attached to microtubules, because the next part is the separation of chromosomes. And that's really important because we need to make sure that all the cells get the correct complement of chromosomes. And that only happens by proper alignment and attachments to those microtubules at the kinetochore. So at any one of these checkpoints, if the criteria is not met for continuing, then the division of the cell cycle at division is going to be shut down. Now, it could just be paused until things catch up, or that process might be shut down completely. If the cell isn't healthy, then the cell will just undergo apoptosis, which is essentially cell death. It's programmed apoptosis is programmed cell death. So it's a process by which the cell shuts down and dies if it cannot continue on safely. Because we're talking about somatic cells, these cellular checkpoints are really important in cancers. So cancers occur due to mutations that cause the cell to basically divide out of control. So very commonly, these cellular checkpoints are related to genes that are going to allow cancer to develop, because when these checkpoints are sort of overridden, you end up with uncontrolled cell growth where cells are dividing and then end up producing tumors because their proper checkpoints are not being evaluated. So let's move on to meiosis. So meiosis is going to be the production of our gametes. So we're going to produce sperm or eggs here. And we'll talk about those processes of actually producing those sperm or eggs in a little bit more detail in a few minutes. But meiosis is the general process that is going to produce those gametes. And in general, it's really similar to mitosis. The overall movements of chromosomes, the formation of the spindles, the lining up at the metaphase plate, the separation of chromosomes is going to be nearly identical. But there are a couple of key differences that allow us to produce those sperm and eggs instead of our diploid cells. So one of the key differences between mitosis and meiosis is that we are going to start at a diploid cell and we're going to produce a haploid cell. Now that's because we're producing these sperm and eggs, and remember, they're going to come back together during fertilization and restore that diploid nature of eukaryotic cells. So in order to be able to fertilize, with each cell having half the number of chromosomes that they need to make an organism, we actually have to reduce the number of chromosomes. And we do that by separating homologous chromosomes. So in meiosis 1, we are first going to separate, we're going to line up our chromosomes, and we are going to separate homologous chromosomes. Now this never happens in mitosis. In mitosis, we only separate sister chromatids. And in fact, that process, that separation of sister chromatids, actually occurs in meiosis II. So meiosis II is where we actually produce our final gamete cells. But at the end of meiosis I, we have reduced our cells to haploid. So we'll go through meiosis 1 and 2 separately and talk about some key features. But the general process is that in meiosis 1, your homologous chromosomes are actually going to pair up. We can see that here. Your homologous chromosomes are actually going to pair up. They're going to do something called synapse, which means they are lined up and attached over their whole length. They're going to cross over, which means they're going to actually exchange some genetic information. We'll talk about why that occurs. And then we're going to continue that process that we saw in mitosis, where our chromosomes line up at the metaphase plate and then are pulled apart in anaphase and eventually are separated into two cells. Again, we separate our homologous chromosomes in meiosis 1, and then we repeat the process again. Our chromosomes line up along the metaphase plate again, except that now we are doing this in two cells. So at this point, when we separate chromosomes there, just like in mitosis, we are separating sister chromatids, with the end result that we produce 4 cells at the end. Now in mitosis, we only produced 2 cells. So in meiosis, we produce 4 cells. Each cell is haploid. So we have 4 haploid cells at the end of meiosis. Now, the other key feature about these cells is that they are each genetically different. So at the end of mitosis, we produced 2 genetically identical diploid cells. Meiosis, we have reduced the number of chromosomes within those cells, and each of them is a little bit different. So let's talk about some of the details of meiosis 1. So in prophase 1, we see the condensing of chromosomes. Those chromosomes line up and our homologous chromosomes pair up. Now they do that by something called a bouquet formation. We'll talk about that in just a minute. Those homologous chromosomes pair up, and then they are physically connected in 2 ways. They undergo something called synapsis, which we'll talk about in more detail, and they cross over, which means they produce this little X-shaped formation there in their pairs, their homologous pairs, and they physically exchange genetic information. Now, all homologous chromosomes have to pair up at least one time. The reason that they pair up is that this holds homologous chromosomes together. Now, this is important because first we have to separate homologous chromosomes, and then we have to separate sister chromatids. So by crossing over and hanging on, essentially linking together, we ensure that all of our homologous chromosomes move together to that metaphase plate so that they can be separated correctly. So that when we get to meiosis II, when we get ready to do that, we can then separate our sister chromatids. So just like in meiosis, our microtubules are going to grow out from our spindle fibers. They're going to attach to our kinetochores at our centromere. They're going to pull them to the metaphase plate, and then they are going to pull them apart. And when they do, again, we pull apart our homologous chromosomes. Now, the result of that crossing over is that we now have genetically different daughter cells. So again, we'll talk about how crossing over does that in just a few minutes. But if you look at these cells, the resulting cells at the end of meiosis I, you see that each of those sister chromatids actually has a little piece of the other homolog. And that's how we get genetically different cells. So let's talk about some of these things that happen in meiosis I, keeping in mind that the process is the same. So we're not going to talk about that process of lining up and stuff again, because we already talked about that in mitosis. Your book actually goes into a lot of detail on this bouquet formation. All I really want to say here is that the purpose of the bouquet formation is that it joins your homologous pairs. So up here at the centrosomes, your chromosomes all line up like you would hold the stems of a bouquet of flowers. So you might use your hands or you might tie a bow around them and hold them together. That brings all of the chromosomes into close proximity. And that's how the cell actually ensures that your homologous chromosomes get together. So when they get together, they recognize, the cell recognizes that those chromosomes are similar and sort of pairs them up. And so you can see down here on the bottom that we've got these homologous chromosomes paired up with their matching homolog. So these are two pairs of sister chromatids that have now come together. So this is the one point in time where your homologous chromosomes actually interact. So we start out with this sort of jumble of chromosomes in our cell. They're all condensed. They're getting ready for meiosis, but they're just sort of all mixed up. So this bouquet brings them together and allows those homologous pairs to associate. So when they associate, we get something called synapsis. Now, synapsis is essentially a process of zippering those chromosomes together. So what's going to happen is if we have, I'm going to draw just one set of chromosomes here. I apologize, my pen seems to be having a freak out. So we're going to draw one set of chromosomes here. These are homologous sister chromatids. And I'm going to change colors just so you can see a little bit more easily what's happening. So if these are homologous chromosomes, we get little protein zippers that are going to form in between. And they are going to keep those in close proximity. So down here, actually we'll go at the top first, you can see if this is one set of chromatids, identical sister chromatids that are produced by DNA replication. And then over here we have our other set. They're going to be joined in the center by these proteins that hold them very tightly together. Now, if you look at this picture down here, this is an electron micrograph of a real chromosome, and you can see that it actually does look like a zipper. If you think about what a zipper looks like on your sweater or your jacket, you can see that here. And it is basically little proteins that are lined up that are holding them together. So this helps to keep your chromosomes together. Remember, that's really important so that we partition them correctly at the end of meiosis. So we have this complex, our book calls the synaptimal complex, and it's just a group of proteins that are going to allow for the synapsis to occur between these proteins. Now, synapsis has to occur before crossing over can occur. So also during synapsis, we have another set of proteins called cohesins. Now, cohesins are sort of ring-shaped proteins that help to hold the sister chromatids together. So we have cohesins that are holding those sister chromatids together, cohesins, and then we have another set of cohesin that's actually holding those homologous chromosomes together. So we have those cohesins that hold chromatids, and we have cohesins that hold homologs. So we've got several different processes that are holding those chromosomes together. So in prophase 1, we need those centrosomes to recognize and pair our homologs. Then we need that synapsis to occur by these cohesin rings that help hold them together, and by those proteins in between those synapse proteins that are holding those sister chromatids together. And then in the last step, we are actually going to perform that crossing over. Now remember that crossing over serves two functions. One is to continue to help hold the sister chromatid homologous chromosomes together. And then the other function, or the other sort of outcome of this, is that we get variety or variation in our chromosomes. So you can see this X-shaped structure here. We're actually going to exchange information between those inside chromosomes. So this process of crossing over occurs in a couple of steps. It looks a bit complicated, but it's actually a pretty simple idea in general. So we start with our, I'm going to start down here on the bottom left actually. We start with our pairs of sister chromatids, so our homologous chromosomes. And they are going to actually line up and produce a break and exchange information between one another. And so this happens by the lining up of your sister chromatid pairs. On one side of each pair, one molecule of each pair is going to be cut or nicked. So we're going to get a single strand break that is going to occur on one side of our chromosome here. So we get our cut here on both of our sister chromatids. Then we are going to join those pieces of DNA. They're going to be pulled together to produce this X shape here. Now that X shape is called a holiday junction. So essentially what's going to happen here is that you're going to produce a break in your strand. So we're going to have two breaks. one on each side. And those breaks are going to be very highly regulated. So they happen at certain locations along the chromosomes. And the number depends on the chromosome. We're showing only one here, two, but at the moment we have one break here. But there are usually any number of crossovers that occur in a particular molecule. So after the break occurs, the other homologous strand is used as sort of a template to repair that break, but it connects the other strand instead. So we have one crossover here, and now we have another crossover here. So we have a second holiday junction here. And what happens here is this holiday junction causes the DNA molecule to kind of bend and twist. And so you actually get this sort of X shape in your chromosomes, and you still have homologous pairing here. So basically, if you were to take this molecule and sort of bend it this way and this way and this way, you end up with this here that looks like a road junction. Looks like a four-way stop, which is why we call it a junction. So then what happens when you sort all this business out is you end up with what were originally your sister chromosomes, or sister chromatids, but each with a piece of the other homologous chromosome. So here, your homologous chromosomes, or I should say your homologous chromatids, have shared information or exchanged DNA. They've shared a piece of their DNA. So you get sequences from one of your homologs now present on the other. And that's where our genetic variation comes from. That's what produces those 4 genetically different cells at the end of meiosis. So let's draw out what might happen. So I'm going to stick with a single color here just because it's easier for me to not switch pen colors. But let's draw our homologous chromosomes. And let's say we have gene A on 1. And I'm going to say that one parent is contributing all of the capital letter genes. But remember that these are homologs. So we have the same genes at the same location with our homologous pair. Now, since we're in the middle of meiosis, we have undergone DNA replication. So here we have one set of sister chromatids. But these two are your homologous pairs. So if we step through what is actually happening in this process of crossing over, we are going to line up our sister chromatids, and they are actually going to form that holiday junction. So over here, if this was our original chromosome on the left, and then the other side is here, so I am indicating that a crossing over event has occurred between genes A and B. So on our other sister chromatid, we're going to draw our A's and B's. Now you might notice that the outside chromosomes are actually not involved at all. These outside chromosomes are going to be identical to the original parents. But the inside chromosomes, these are going to be the ones where the crossover exchange has occurred. So these two in the middle are actually going to share genetic information. So when we separate sister chromatids, we separate homologous chromosomes, we're actually going to see where we have exchanged that information. oops, I'm sorry, I have a mistake here. This one should be the capital still. So the parent one, the crossed over one with the lowercase genes there. So this one here, we're going to see the opposite crossover. And we end up with this outside chromosome being the same as the parent, and this inside is the one that has exchanged. So if we were to separate these out, which would not actually happen, but you can see, I'll draw them close together so we're still in our sister chromatids. You can see where the differences are. Sorry, that one is still the capital. I keep doing that. And then our other sister chromatid is going to be the opposite. So when we separate these, we'll end up with 4 daughter cells. Each one of those, after we go through this process of meiosis 1 and 2, each one of those is going to be a different daughter cell. So you can see we have new combinations of those A and B genes. So then after meiosis 1 is complete, after we have produced our 2 daughter cells, we are going to then each of those cells enter meiosis 2. So this is just indicating that we had 1 cell. It was divided into 2 cells. This was our 2N cell. And remember, at the end of meiosis, we have reduced our cells to haploid. So here at the beginning of our meiosis 2, we are going to be haploid from here on out. So we're going to separate sister chromatids in this process, and it's basically the exact same process as in mitosis. So meiosis 2 is identical to meiosis 1, where we line up our chromosomes along the metaphase plate. We pull apart our chromosomes in anaphase, and then in telophase and cytokinesis, we begin to split our cells, reform our nucleus, and then the chromosomes decondense. So at the end, we have reduced our diploid cell to haploid, and we have produced genetically different cells, and we have produced 4 cells. So we get our 4 haploid genetically different daughter cells. And you can see that genetically different here. If you look at the colors that make up these chromosomes, we have this blue chromosome has a little bit of its homologue. Purple has a little bit of its homologue. And each one is going to be a different combination. So before we finish up with meiosis, I want to sort of recap the importance of meiosis. The reason that meiosis is so important is twofold. One is that we reduce the number of chromosomes. So we produce our haploid cells. But the other is that we get genetic variation at the end of meiosis. Now, this variation comes from a couple of different places. One is the way that the chromosomes line up at the metaphase plate. So if you were to look at these 3 sets of homologous chromosomes, you might notice this one is sort of backwards. They have drawn red and blue instead of light and dark. And that's, I think, to indicate like mom and dad, like dad gives the blue, mom gives the red. So they have indicated their chromosomes a little bit differently. This is from a different book. But the way that those chromosomes line up is going to give you some of the variation that's produced in meiosis. So we have lined up here red, red, blue on the top, red, red, blue. And on the bottom, we've got blue, blue, red. Now that means half the cells are going to get 2 of mom's red chromosomes and 1 blue. The other half is going to get the opposite, 2 blue and 1 red. Now, these chromosomes could have lined up in a different pattern. If this one in the center was lined up the other way, we would get red, blue, blue, but different combinations of red and blue. That means on the bottom, we would get blue, red, red. But it would be a different combination of chromosomes. So there's one way you get variation. The other is that process of crossing over, where we've actually exchanged some information. So not only does the combination of mom and dad's chromosomes at the metaphase plate and the way that they're oriented produce variation, but then we get that crossing over on top of that. And then finally, after meiosis, we get another important bit of genetic variation. And that variation comes from the fertilization of a random egg with a random sperm. So if you think about 2 individuals, one producing sperm, one producing egg, they should be unrelated. Remember that even though humans have similar genetic information, those homologous chromosomes still have differences. And those differences are enough to produce variation in the offspring. And so when you get fertilization, you have a random egg with a random sperm, and those homologous chromosomes contained in each one is also different from one another. So we have genetic variation in meiosis coming from 3 different places. We have the arrangement of the chromosomes in at the metaphase plate. Then we have the crossing over that shuffles bits and pieces of parental chromosomes. And then when that gamete that was produced from meiosis is fertilized with another gamete, you're going to get even more genetic variation. And although we don't talk about it too much in here, the actual process of evolution, variation is really important for evolution. And that variation that from meiosis is really the only new genetic variation that comes out of a population. It's sort of the reshuffling of genetic variation. So before we move on to sperm and egg production, I wanted to talk about how we count chromosomes, because sometimes students are a little bit confused on how we actually determine how many chromosomes are present in a cell. So we count chromosomes by the number of centromeres. So this is Meiosis II. We have centromere, centromere, centromere. We have 3 chromosomes. Now, I realize that we have 6 pieces of DNA because each of these chromosomes is made up of a sister chromatid, but we actually only have 3 chromosomes if we count number of centromeres. So 3 chromosomes because we have 3 centromeres, because they are attached at the centromeres. Now, when we pull them apart in anaphase, very, very momentarily, we actually have 6 chromosomes in this cell, but the result is 3 chromosomes. So even though we have sister chromatid pairs at the beginning of meiosis, we are not actually reducing our chromosome numbers there because these centromeres hold them together and make them 1 chromosome. So back in our original cell, this is going to get really crowded. Oops. We actually had, let me change my color. When we were still diploid, we actually had 1, 2, 3, 4, 5, 6. 2n equals 6, even though those chromosomes are made up of sister chromatids. So then when we reduce down to n, we again count our number of centromeres, count our number of centromeres, and that is where we are reduced down to haploid.